Yep. Thanks, Duccio. So, I mean, he already said everything. So, hi, thanks for having me. So today I'll try to talk about some works that we did in collaboration with the team at Broad regarding the use of deep learning techniques in biology, especially in genomics. I believe all of us are physicists, right? I'm a physicist too, so disclaimer, I might say something biologically inaccurate. I'll try to do my best to keep it like for my limited understanding of biology. But I think this is interesting because the fact that we have at our disposal machine learning techniques and like a solid mathematical understanding on our side as a physicist allows us to build pipelines, algorithms and stuff which are useful in domains which are not necessarily like, I don't know, deep physical stuff like high energy or astrophysics, as Lorenzo and Matilde told us yesterday, but we can also go a bit outside of the normal tracks and work in biology, which prior to this work, I knew absolutely nothing about. I mean, level of high school stuff. Now I know absolutely nothing, plus something that I needed to learn to make this machine learning stuff work. But it has been like really interesting. And I think there are a lot of lessons that one can bring back to physics, because in the end, we are always analyzing images, we are always analyzing data, we are always dealing with stuff. So, I mean, it's always nice to go to a different place, do stuff, and then come back with some new knowledge and some new sense of how can one do with the tools that we have. So the collaboration of this work is huge. Like, there have been a lot of people who have been collecting data in various experiments in the laboratories at Broad of Aviv Regev and Eva Makosko. Mainly, this collaboration was made possible by Tommaso Biancalani, which was like many years ago was at the University of Florence, and he kept in touch with Duccio, and we managed to pull off these collaborations. And each one of us had a tiny little part in this big puzzle that I'm gonna tell you. And so these are all the people that mainly collaborated at the work in various forms. Okay. And also like the various fundings and stuff which we don't care about really. Okay, so what is the biological background of all of this? Very hand-wavily. Okay, so the background of this is the Human Cell Atlas project. What it is, this Human Cell Atlas is basically a big endeavor which comprises many other sub projects like HubMap, BCCN, and the Chan Zuckerberg Initiative, and many other like the Broad Institute, etc., which is basically the idea of building like a Google Maps of the human body. Okay, so from the level of the whole body and organs to the level of tissue to the level of single cells, the idea is building a comprehensive framework that can tell you what's inside us, basically. Okay, and so you might want to have 3D models of organs, so like these ones of kidney, this 3D model of kidneys from HubMap. Or you might want to have have information about tissue. So this is a piece of cerebral tissues where the lights are neurons and their connections. So the fluorescence is emitted by neurons and the connections from the BCCN project. Or you might want to explore the manifold of the transcriptome. Okay, so each one of these points is a single point in the RNA transcriptome space, so the space of all the genes. Okay, and this is a two-dimensional representation of it. And you might want to segment this manifold for cell type. So maybe this cluster is a certain type of cell, this other cluster is another type of cell that exploit different functions, okay, inside the same tissue. Okay, so you have different scales. Okay, and what you want to do is go from the whole body to all the way down to the single cellular information. Okay, as I showed you. This thing, since you want to build this comprehensive framework, you want to kind of connect all these scales. Like you want to know where like an organ is inside the body, you want to know where a certain type of tissue is inside an organ, and you want to know which cells are where inside the tissue. Okay, so in a way that you can connect all these informations that you get all in one place. Difficulty is most often you collect this type of information using different like specimens, different technologies, different like everything, right? I mean, at the organ level, you can just do an autopsy and say, hey, this organ is here. Okay, at the histology level, you have to take out the organ, slice it up, okay, stain the things, take images of that and reconstruct the organ in 3D. At the cellular level, you have to do single cell RNA sequencing and stuff like that. And so you have entirely different technologies, and then you have you need to have a way to recombine them together, okay, in some way. And this recombination is the thing that I'm gonna talk about in more detail. It was the focus of our work, okay? So let's move now to the mouse brain, okay? I told you the big picture is this human cell atlas thing. We go one step back and we go to the mouse brain. So of course, we have a lot more information about mice in general than we have to human, because we can do a lot more stuff with mice that we cannot do on humans due to ethical stuff and et cetera. So we have a lot of information, and we already have an atlas of the mouse brain, okay? So it's just one organ of an organism which is not human. So, I mean, there's a long way to go, okay, to have a complete human cell atlas, but that's the beginning. So in the mouse brain, we have a common coordinate framework. So we have a set of coordinates, okay, in which we can upon which we can superimpose different types of information. So, okay, for example, we can superimpose ontological information about like information about the ontology of the tissues that we have in the mouse brain. So here, different colors represent different types of tissues inside the brain. We can superimpose information about the connectivity of the various neurons inside the mouse brain. We can superimpose information about the cell position. So we can have a cell density map and say like how many cells are in a certain volume, so where are they located in this common coordinate framework. And all these like things here are data sets that you can actually go and look up to I don't know, if you are interested in the connectivity of the mouse brain, if you are into network science and you want to do a project with a real kind of network, you can look up this. Like there is a lot of data, okay, about this stuff, and this is why we started to use machine learning to tackle this kind of problem. So what we are missing single cell data. So we have now recently reached the ability of sequencing a high number of genes from a high number of cells. So we can take whole pieces of tissue and sequence them at a single cell level with a throughput of roughly like 30,000 genes. Okay, so we have like, we are able to extract a lot of information from single cells. Okay, so these are data collected by the labs at Broad, whom we collaborate. The problem is we have no way, okay, of since like this is very new stuff. Okay, this the ability of having single cell RNA sequencing data. We lack the ability of mapping this information in the common coordinate framework. So we know that we can take a piece of tissue, we can sequence the cell. The problem is that sequencing the cell means actually destroying the tissue, so destroying the spatial information, okay, that we have. And we have no way of saying, once we have the single cell sequencing data of the genome, we have no way of placing it back into this common coordinate framework and have a spatial information about how genes are distributed, how cell types are distributed. We can just say, okay, we have a certain density in this whole bunch of tissue, but we want more, okay? So, for example, here is like the sequencing of the primary motor area of the mouse brain. Okay, so this the primary motor area is this one highlighted in red, and this was a work that was done prior to ours, which we heavily leaned on for our work, in which they take samples of this tissue, they destroy them to do single-cell RNA sequence, and then using UMAP, which is a dimensionality reduction, which is very popular, I believe you all know it, they kind of plot in 2D the manifold of these 30,000 genes, okay, that they obtain, and using that they can like identify 22, if I remember correctly, the number 22 different types of cells that you find here color-coded and with the names on. I have absolutely no idea what these names mean. I have absolutely no idea what these cells do, okay, more or less. But okay, this is a first thing that you can do. So you have this huge manifold, okay, you reduce it to 2 dimensions, and then you can segment finding different cell types, okay? Now you know that in this tissue you have some cell types, but again, you are missing the information of how are they spatially distributed, okay? So let's now talk about why we cannot spatially resolve whole transcriptomes at single cell resolution. So what I've talked to you is this single cell or single nuclei RNA sequencing techniques, okay, which are these techniques that physically take the tissue, destroy them in some solution, and then cell by cell, they sequence the whole transcriptome of the RNA. But they lack the spatial resolution because they physically destroy the tissue. There are some other techniques like MERFISH or StarSec, I believe it's called, which are called Insitu Techniques, which are techniques that rely on imaging of parts of the tissue. They can go at single cell resolution, but they lack the ability of sequencing the whole transcriptome. So they are usually limited around a few hundreds to thousands of genes instead of the whole transcriptome, which is roughly 30,000. Okay, so they're very limited in the bandwidth, okay, that they can achieve, but they're very precise. They go at single cell level. And then we have spatial transcriptomics techniques, which is a huge branch. We will use mainly the Visium techniques, which they have a high throughput of genes. I believe they can do the full transcriptome, but they lack the ability of going at single cell resolution. So they have a limited resolution, and they are subject to dropout genes. Okay, so due to technical stuff, black magic, sometimes they kind of skip some genes in the transcriptomes. Okay, randomly. So you see that all these techniques, they all have advantages and they all have drawbacks. Okay, what we want to do is to build, okay, a deep learning stuff, okay, which I will explain to you, that can take the best of all these words and basically harmonize all these data together in a way that we can build a whole transcriptome map at a single cell resolution. Okay, basically that's it. How can we do that? So we do this in 2 parts. The first is to integrate the single cell RNA sequencing data with spatial data like MERFISH, okay, the Insitu techniques that we got. And then when we have integrated single cell RNA sequencing with spatial data, we can integrate the spatial data with the histology, okay, and so map it into this common coordinate framework and have the full spatial information. The thing that I'm going to talk here is you can find all this in this paper that we did. And this first part was the contribution of Gabriele. This second part was mainly my contribution. So yeah, just for acknowledgement. So let's start with mapping. So the first part. So we want to map the single cell RNA sequencing data into the spatial one. So what we have is this: we can collect single cell RNA sequencing data from healthy adult mouse brains. So we take the mouse brains, we slice them up, okay, and then we take a punch, they call it. So we take a sample, okay, of tissue, and we have like 3 regions of interest at 3 different like depths, okay. So from which we collect single cell RNA sequences. So these tissue samples are collected, destroyed, and sequenced. we are able to sequence 160,000 cells, and we can, again, cluster them, okay, into 22 cell types, okay? And we have, for each one of these cells, we have the whole 30,000 genes of the transcriptome available. Then we can take these single cell RNA sequencing and try to map them into MERFISH data. So MERFISH is this techniques that is in situ and relies on imaging, which is able to map small portions of the tissue. So this portion here would be like a sub portion of this region of interest here, is able to map small regions of the mouse brain. So this in particular is a piece of this primary motor area here. And these dots are single cells, okay? But is able to deal with only small regions, so 4,000 cells, and with a low bandwidth of genes, so 254 genes per cell, okay? So what we want to do is to take our high bandwidth data and map it into this bandwidth data in a way in which the gene distribution is consistent, okay? And in this way, we hope that we will be able to increase the bandwidth of this technique, okay? So how do we do it? So let's introduce a bit of notation. So I will use different indices. So index I is for a cell, okay? So for a sequenced cell, of which I have the transcriptome, and I have a certain number of them. I voxelize the space at the finest possible resolution. So in this case, each voxel is spatial, like volume that I'm mapping on is a single cell, okay, because this technique is a single cell. We will see later cases in which the voxel is actually comprises of more cells due to the limitation. And the index K is for genes. So I have a certain number of genes that I'm able to sequence with MERFISH, 254. And so this index will have dimension of n genes, okay? So what I get from these two sets of data that I have is a matrix S from single cell RNA sequences, which has a dimension number of cells times number of genes. So it tells me each cells which genes expresses in what percentage, okay? And I have a matrix that I'm going to call G, which is the one that comes from the spatial technology, in this case, MERFISH, which has a dimension n voxel times n genes. So it is going to tell me in each voxel of space which genes do I have, okay? These 254 ones, okay? Plus, I have, since I am able to resolve these voxels as a single cell resolution, I have also a vector. I can extract a vector of cell densities, okay? And what I want to learn is a mapping matrix, okay, which will have a dimension of n cells times n voxels, which tells me the probability of finding the cell I, which express the genes K in the voxel J, okay? If it makes sense, okay? So, yeah.
I'm assuming that either amendments and was structured in the SMG or
no. This is what we are gonna learn. Okay. Yeah, the mapping matrix is what we want to learn. Okay, what we do have from the single cell sequencing data is the matrix S, and what we have from MERFISH, the in situ technique, is the matrix G. Okay. Okay. So I want to find a mapping matrix M. I'm gonna show you, so that the product of M times S, so the spatial, oh, sorry, I skipped. So the spatial distribution of the genes on the voxels is as similar as possible to the gene distribution on the voxel that I get from the spatial technique. Okay, so that's the aim. So we formulate this problem as a minimization problem, okay, and we minimize this objective function using PyTorch. So now I'm calling M tilde my matrix that I want to optimize, because I want to use the softmax of it to ensure the fact that this mapping matrix is constrained between 0 and 1, and the columns sum up to 1 because they are probabilities. Okay, and I have three terms in this optimization function. So this first term is a Kullback-Leibler divergence between the cell density vector, which results from the mapping. So my mapping will predict a certain cell density distribution, and I want this predicted cell density, this mapped cell density, to be as close as possible to the real cell density that I measure from Merfish or whatever. Okay, so the cell density has to be consistent. This other term tells me, so this cossim is the cosine similarity function, which tells me basically that for each gene, okay, this sum goes through the genes. For each gene, I want the spatial distribution over all the voxel to be proportional to the one measured by the Merfish techniques. And these other terms tells me that for each voxel, I want the gene distribution to be as close as possible to the measured one. Okay, so it's basically just a probabilistic mapping, which ensures that what I'm mapping from the single cell RNA sequencing in space is consistent with the density and the expression of genes at the cell and spatial. Okay, makes sense? Questions? Okay, so we optimize this using PyTorch, and we find this mapping matrix M. So what we can do now that we have this mapping matrix? Well, we can take the single cell RNA sequencing data, on which I showed you that you could identify each cell, which type of cell it was, okay? And we can build a map, okay, of cells at single cell resolution. So I can map using different color, and each one of these color codes a different type of cell, okay, in space. So I can see how different cell types are arranged in space, and I can build a probability map like for a certain type of cell. For example, here we have different types of excitatory neurons, and I can see how they are distributed in space. Okay, and this is cool because I can see, oh, here I have a very high density of excitatory neurons. So I can ask why, and I don't know. Then it's biology work, and I don't know what I want to do with it, but it's cool. Okay. The other thing that we can do is the other thing that I want to be able to say is the following. I want to really trust my mapping. Okay, so now I've optimized this thing on the 254 genes. 254 genes that I had, and I'm kind of sure that for these genes here, my mapping is correct, kind of, because I've like minimized over it. But how can I know that for all the other genes, the mapping is kind of makes some sense? Okay, so I have to kind of validate that for the genes that I haven't explicitly mapped in my cost function, I can call them test genes if you want, my mapping is kind of faithful. So I can rerun this. Oh, actually, I forgot to tell you why we call this algorithm Tangram, because it was reminiscent to kind of the Chinese puzzle game in which you have to arrange triangles all together, which is called Tangram, and this is similar to what we're trying to do. So we have this information and we're trying to arrange it to space. So it was a cool name, and we named it Tangram, this mapping algorithm. So how do I validate the holdout gene, the test genes? So first thing, I can rerun Tangram, okay, the mapping, but holding out, in this case, 9 genes. So instead of mapping on 254, I map on 245, okay, genes. And I check that the predicted distribution from Tangram is consistent from the one of Merfish that I know because I have them. And you can see that I have, like, this is pretty consistent. So here on the x-axis, I plot the spatial correlation, but the more sparse the genes are, the less, like, the worse is the correlation, okay? So, but this gives me a good idea of how much I can trust my kind of mapping algorithm. Another way that I can do it is by by confronting it with other atlases. So for example, here we have the Allen Atlas, okay, which basically has collected various genes, not the whole transcriptome, but a lot of genes which are not mapped in MERFISH by staining histological images for a certain genes. So for example, these genes, this NOS1AP, okay, is the expression of this gene is corresponding to the stains in this image. So the ink is telling us where this gene is more expressed. And you can see that is expressed in this low band of the primary motor area, which is the same of the one that we predict from our mapping, okay? The same you can see here is even in this case is pretty clear. This ATP gene is expressed in this low bands region, which are at the edges of the primary motor area. And again, we do predict with our mapping algorithm that these are expressed with more density at the edges, etc., etc. You can see, I don't know, other cases here in all these cases, but we kind of validated with like holdout genes and with other atlases and information that we had on the mouse brains that the mapping that we learned kind of made sense even for genes that were not explicitly mapped, okay? So in this way, we can confidently say that we can increase the gene throughput of these in situ techniques to 30,000 genes, okay? So this is the first result. Now, instead of being able to express just to map in space at a single cell resolution just 254 genes, we can map the whole 30,000. And that's the first result. So the other thing that we can see is that we can kind of correct low quality genes. So this is a mapping performed this again Tangram, but instead of using MERFISH as a reference dataset to map on, we use this star map technique, which is an analogous in situ technique. The difference between MERFISH is the throughput of star map is a bit higher, okay, in terms of genes. So now I have 1,000 genes, but to achieve this throughput, I have some genes randomly that have very low quality of samples. So I'm going to call these dropout genes even in the following. So for example, you have these genes here, which they have very poor quality on star map, whereas on Tangram, these genes are kind of automatically corrected. And you can see, for example, here for this FAM gene, the Tangram predicts that the expression is concentrated in the middle of the primary motor area, and you can see from the stained image that indeed the stain is concentrated in the middle. So our mapping corrected this low quality sample of star map, okay, which is another cool feature to have. Okay. Okay, questions on this part? Yeah.
Is there more statistical? How long we're doing?
No. Okay, that's the point. You kind of have at a certain point you have to trust that since it works well on the set that you were able to test on, it will work well also on the others. You do not have 30,000 stained images for all the genes. You have I don't know how many you have, to be honest. You have some of them. But yeah, I mean, you have to compare what you can compare, and for the others you have to trust it. There will be cases later where we have some more information and we can do a more accurate statistical analysis on what is going on, what is going well and what is going wrong. But in this case, no. Basically, you say, okay, for the genes that I can control over is mapping kind of well, so it's going to map kind of well for also the other genes that I can control over. Yeah. We have compared all the stains that we had. And again, you can build like the measure that you can use is a spatial correlation thing. And it gives like good values. For the stained images, I'm not even sure that you can do a spatial correlation function because I'm not sure if these are single cell resolutions, but I believe that for the stained images, the best you can do is kind of a eye comparison. You say, hey, here I have the concentration, which is the expression, which is concentrated in the middle, and the stain is concentrated in the middle. well, that's good enough. Okay, but that's it. Yeah. That's. I mean, you are limited on because you have different type of data, different resolution. You have a lot of constraints that you have to work on. And also, you will see when we go onto the more deep learning stuff, though this was a probabilistic mapping, the fact that you have a low volume of data taken across like different types of technologies, etc. When you go to train stuff, it's a problem. It's a pain in the ass. But I'll come to that in a minute. But that's the nice thing about working with real data. I mean, you encounter a lot of problems which you never think about when you are using, I don't know, simulated data, or I don't know, you build a machine learning model using the usual, I don't know, CIFAR10 or MNIST benchmarks, that everything looks fine and cool. And then you go to the real world and you have like 20 images taken with a very low quality, and you say, okay, now what do I do? I mean, okay, well, it's fun. I mean, to me, at least, that's fun. Okay, that's it for the mapping part. Okay, now that we covered the mapping part, we go to the second part, that we take the spatial information and we want to integrate it with the histological images. Okay, so what is the problem here? That I have an experimental stain image. So this is a slice of the brain taken on around the ZY plane. Okay, so it's a vertical slice here, and I want to know the posterior coordinate, so the x coordinate, or the depth, okay, on the brain at which this image was taken. Okay, so depth 0 will mean the olfactory bulb. Okay, this is the olfactory bulb, and x, I believe it was like 13,000 microns, which is like 1 centimeter and something, is the cerebellum, okay, in the back of the brain. I want to know at which point was this slice taken. Okay, and to do this task is called image registration. Okay, so I have this reference atlas, which is the ideal mouse, the prototype of the mouse brain. Okay, let's call it this way. And from this atlas, I can call a lot of information. I will show you later. Like, yeah. This is kind of the reconstruction of the prototypical mouse brain. So they have collected a lot of specimens, mapped the histological. It's kind of the average mouse brain. Okay, that's the prototype of the mouse brain, and inside it you can call a lot of information. Like you have the cell density information. You have like all the various mode of data that I showed at the beginning, like the connectivity information, the cell density, the tissue information. You can all extract them from these reference atlas. Okay. So what we want to do is to register this image in the reference atlas. So find the correct match. Okay, for example, this is the analogous. You can see, you can find these features. So you can find these cavities here. You can find this split in the middle. I don't know. You can convince yourself that these are the same piece of the brain. Okay. How is this usually done? Okay, is done with human supervision. So you have to take an experimentalist that has to identify some landmarks in the image. So for example, the cavity here, this split in the middle, I don't know what they look at. Okay, they are biologists, they know the stuff. And then you have a program which is called ANTS that kind of maps this image into, like, registers this image into this other image. Okay, automatically given these human landmarks. But this process takes time. Okay, and we want to build a thing that is kind of automated. Okay, from top to bottom. So this registration, we want to have it all like automated. So how can we automate this registration process? To do that, we use the face recognition techniques, because this task of having a reference database and having a new image and having to check whether like the correspondence in the database is very similar to face recognition. For example, if you are, I don't know, a company and you want to face ID your employees when they come into work instead of having them badge, you need to have a database of reference employee image, one for each employee. And then each morning when they come in the office and they're dressed a little different, and they have glasses or not, or they have a different hairstyles because they have been to the barber the day before, etc., etc., the deep learning algorithm has to still recognize the employee. So the task is really similar. Now I have an experimental image, which is all like has all the noises and all maybe some parts are cropped out, maybe slightly tilted, etc., and I have to match it with the reference image of the atlas. So the task is really similar. So we adopted the Siamese neural network model, which is commonly used in face recognition, in which we have the Siamese part is this one, in which we have two images, which are pre-processed by some shared networks. Okay, so these two are actually the same networks with the same weight in the layers. And we used a pre-trained part, and then we fine-tuned the last convolutional layers plus a couple of fully connected one. And what we did with this embedded the image that we passed into a latent space of dimension 512. Okay, and in this latent space, basically, I have the representation, okay, of this image, okay, which will hold some meaning with respect to the depth of the brain at which I'm looking at. So what I want to find is at which depth was this experimental image taken. And then what do? I take this latent space embedding that I learned, I compute the L1 norm, and I learn a similarity metric, which in our case is the actual distance in micrometers between these two images. So let's say the experimental image was taken at 700 micrometers of depth, okay, in the brain, and the atlas image that I'm comparing it to was the one taken at 500 micrometers, okay? The distance that I'm trying to predict is 200 micrometers, okay? So I'm trying to actually predict the distance in depth in the brain between these two images using the latent space information, okay? So I trained this thing. So these are the details. So for the pre-trained encoder, so this convolutional part here, we used a DenseNet 169 pre-trained on ImageNet. Okay, yeah. Yeah, between two images. So I have this like a pair of images which go through the Siamese, so through the same network, okay, and in the end they will be embedded in the latent space. Then I get a scalar. Yeah, no, this is L1, so it preserves the dimension. This is L1 norm, so this will be a 512 dimension. Yeah, okay. And then I feed it onto one in one layer that will predict the actual cortical depth. Yeah, okay, makes. Yeah, the first part kind of represents the image in a way which is like in a way in which it kind of identify the distinct features of the brain. So it will be analogous to finding these landmarks, okay, finding the distinct feature that tells you at which depth you are looking at, okay, removing all the experimental artifacts and stuff. And this one is actually predicting the depth, okay, if you want to have an. Okay, so the pre-trained part is a DenseNet, pre-trained on ImageNet, so is a dataset which is really different, like from these images that we're looking at, because ImageNet are like images of birds and horses and cars and stuff like that. But it's useful because it can extract like features, like high-level features, like, I don't know, round corners, shapes, stuff like that, which is nevertheless useful. And then we fine-tune the last convolutional layer plus the two fully connected layers that we add just prior to the latent space. And we train the thing for 50 epochs using 1800 image pairs, okay, per epoch in batches of 16. And the loss function that we used is a simple mean square error because we just want to compare the two distances and at the end, so that's it. What we needed to do is to have heavy augmentation, use heavy augmentation for training. So we started by training like really vanilla with the images that we had. And we noticed that the thing wasn't really working because it was picking up a lot of these artifacts. So like the fact that we have these cropped things, so you can you can maybe, I can show different, okay, I can show these that we have different, so some of them have different backgrounds, they use different stains, okay, so this is the ref, okay, here is better, so I am gonna come back, but you can see that different images taken from different labs use different stains, they have different backgrounds, some of them have like missing parts, they have holes punched in them because maybe the group collected some tissue to do, I don't know, single cell RNA sequencing and stuff like that. And these artifacts were really like the deep learning thing was going crazy without augmentation. Okay, what we did is to use like heavy augmentation. So we cropped out like parts of the images. We applied random rotation. We applied some deformation. We applied like random background shift. We added some noise. Like we threw all the tricks in the book to do data augmentation, and it kind of worked. Okay, in the end, and how we were able to tell that this works. So this is a kind of an easier task because we were able to hold out some images for testing, actually. And so the first check that we did is on the latent space. So we take the 512 dimensional latent space and we plot it using UMAP again, so the dimensionality reduction technique. And what we see is that the structure of this 512 dimensional manifold is actually a 1D structure when the meaningful variable is actually the depth of the brain. So this color code is from 0 to 13,000 micrometers, so the depth of the brain. And you can see that this one-dimensional manifold is nicely ordered from 0 to 13,000 micrometers. Yeah. Random. No, we picked the random pairs. So it was not necessarily experimental versus Atlas all the time. Exactly. No. We did it randomly. So we picked 2 images at random, and we wanted just to predict the difference in depth between them. Okay, so that's the task. And now here I showed the Atlas images because it was in the end, we at testing time, what we're gonna do is take one experimental image and compare it with all the Atlas images and find the one that has the minimum distance. Okay, and that's our best match. Okay, so first thing is that we see that the latent space is a nice one-dimensional structure, okay, which is cool because it means that it kind of learned the meaningful variable, which was the depth, okay, which is the one that we are interested in. And these points are the same, they slice it as the same depth taken from different data sets. So these are a couple of experimental images taken from different groups, and these are images from 2 different atlases. Okay, you can see that we also have different atlases. So this average mouse brain, you can compute it in different ways, and you see that computing the average mouse brain in different ways will result in slightly different stuff. I don't know, they have all their theories of which one is better, but I'm not going into detail on that. And you can see that points that are actually close in depth are close together, okay, in the latent space, kind of all nicely and grouped. Okay, so for example, now what we do is we take a new image, okay, so these are test images, and we call, we use the model prediction to do this depth calling. So we take one experimental image and we scan. So we pass it, we confront it using the Siamese neural network with all the atlas. We take the minimum distance, and that's our match. Okay, that's our best match. And you can see that indeed, even with images that are really like, they have really low quality of sample because they have all these missing pieces, etc., you can convince yourself, even if you're not a biologist, that this is indeed like kind of the correct reference. Okay, also for this one that has these holes in it, like the reference is pretty good, so you can see these kind of cavity features here. You can also, in this image, which has a lot of noise inside, you can kind of make out these lobes these lower lobes here, and you can convince yourself that actually this reference is correct. How we validated it, we took it to a biologist and we said, do they look good? Okay, so yeah.
How's the distance for the distance? So if you give the whole scout, it should be like a...
I'm gonna arrive. So the next thing that we wanted to do is to also assign a confidence of prediction to these kind of things, because sometimes we saw predictions that were not this nice. Okay, this is a nice sample. So what we did is exactly what you were suggesting. So we predict over the whole depth, okay, from 0 to 13,000, okay, and each one of these blue crosses is the prediction of the model in distance. The minimum is our best thing, our best guess, our best depth calling. And then we try to fit it with a piecewise linear function, which is what we would expect, given that this is just a linear depth, okay? And we can use the quality of fit, the goodness of fit, as a measure of a goodness, like of the confidence of our estimation in depth. So you can see that for these two cases in which the fit is pretty good, you can see that the depth calling makes kind of sense. Okay, here they're pretty much the same. So this is a very good image. This one, even if the experimental image is tilted, it's able to retrieve, like you can convince yourself that these two are the same one. But this one, in which predictions are all over the place and the fit is really not that good, like you cannot find a piecewise thing that fits well this data, you can see that actually this assignment makes no sense. Like you are missing all these lobes here that are not present in the experimental image. So this is kind of completely misplaced guess, okay? But luckily with this technique, we were able to kind of find a confidence score and say, hey, if the confidence is under a certain threshold, ask an experimentalist. Do not call the depth yourself, okay? This needs human supervision, okay? Which is a nice thing to have. Okay, questions on this part? Okay, so the next thing that we want to do is to perform anatomical region calling using semantic segmentation. What does it mean? It means that taken the experimental image, we want to segment the various tissues that we have. We have 5 types of tissues. So okay, we have background, which is not type of tissue, but and 4 other types of tissues, which are cerebellum, cortex, general gray matter, and white matter. And why we want to perform this image segmentation? So we want to go from the experimental image to the color-coded one, because these segmented images will serve as a replacement for the manual annotations in the registration pipeline. So now instead of, I'm going back a couple of slides. So instead of using ants to register the images using this landmark will be these segmented regions, and we are going to use ant to match these to register these segmented regions. Okay, so we are using the same program, but now these landmarks are gonna be like automated. Okay, kind of. Okay. So to do this segmentation, we used a UNet architecture. Again, this was, I believe, so this was the work of Ziking and Aman, and I believe it was like some layers were pre-trained on ImageNet as well, and then we fine-tuned like the central part of it to perform this thing. This segmentation part didn't need to be very precise, okay, because we already have the depth calling, and we are going to use these segmentation marks just as a guide for the registration. Okay, so it doesn't need to be super precise. It can be like, it can have like rough edges, and it can misplace some portion of the tissues. It's not a big deal, as long as it segments well most of it. The registration software will do a nice job of it, because usually it works on a single point. So you are providing a lot of points. So it's much more than the software needs, anyways. So with this UNet architecture, we are able to produce consistent masks, okay, for images, even like, again, with missing parts and stuff like that. So we can kind of recognize well these various types of matter, which basically are different colors of stain, more or less, okay? And so now our complete registration pipeline is the following. So we have the experimental image, and the first step is that we use the Siamese neural network to call its depth in the atlas. So we say, okay, this image was taken 700 micrometers, okay? And then from the atlas, we have the mask of the atlas, which is inside the atlas. So it's inside the database. I can call it exactly, okay? And then I can use the UNet to produce the mask of the experimental image, okay? And now I have these two masks that I can register on top of each other, okay? Again, having these two masks be registered on top of each other is the same of having this human annotated landmarks, which this ANTS program was registering on top of each other, okay? Makes sense how this pipeline works? And so now what we have automated is the following, is that from a certain like image, okay, we are able to extract a lot of information from the region of interest that we want to perform mapping on later, okay? We can extract, for example, the anatomical region map, so understand which type of tissues of different like tissues are in each part of this region of interest. We can extract the cell density map. We can extract all these other information that are embedded in this common coordinate framework, okay? And this is the beauty of having a common coordinate framework and a consistent atlas, that you can retrieve a lot of different type of information just like from a single place, okay? You don't need to jump back and forward against different coordinate system, different measurement stuff. It's just you registered it and you are able to search through the database for a lot of information. Now that I showed you the cell density map, you kind of know where this is going, because the cell density was one of the things that we used to map in the Tangram mapping algorithm. So we are going to part 3, which is mapping of spatial transcriptomics. So now we have registered this image, and we can use the information that we can extract from the common coordinate framework to map, again, the single cell RNA sequencing data on spatial transcriptomic techniques. So the spatial transcriptomics are these other family of techniques which have a high throughput of genes, but they're not able to go at single cell resolution. They resolve these voxels, okay, which comprise of, I don't know, a couple to 10 cells at most. Like they are small, but they're not single cell resolution, okay? So again, what we can do using this data set is, since we have the whole transcriptome now, we can fit the gen expression, so use Tangram, the mapping that I showed you before, on 1,000 marker gene, and then actually validate it on the others, because now I have all 30,000, but I'm training only on 1,000, okay? So, for example, so here are the results. So these are just the results of the mapping algorithm. So these are the distribution of excitatory neurons. These are the distribution of inhibitory neurons, and these are the distribution of non-neuronal like stuff of some, okay, of sometimes types of cells, which again I don't know what they mean, the specific names. On this region of interest. But what we can also do is the following. So we can, inside of each voxel, okay, now I have a gene distribution inside of each voxel, and using, again, the automated, like the pipeline, I can exactly know how many cells are in each voxel. And I can kind of predict probabilistically, given the transcriptome that I have inside the voxel, which cell types are most likely to be inside of it, okay? So, for example, I have, I don't know, in this voxel, I have 4 cells, and I look at the whole expression of genes that I have into that, like into that voxel, due to the mapping, okay? And I can try to kind of predict probabilistically which types of cell are more likely, so for which combination of 4 cells is more likely to give me that transcriptome in that voxel. Again, This is very uncertain, okay, this mapping, because there are many combination of cells that can give you roughly the same transcriptome, but at least is kind of a first guide to go to guide you to single cell resolution in these techniques, okay, which are not natively single cell resolution, okay. Again, we have in this case, we have absolutely no way of testing this because we have no match to tell us if the assignment that we made kind of made sense or not. It's just a bona fide stuff that you believe that if the combination of cell that you have selected reproduces the transcriptome that you measure, there is a good chance that the guesses that you're taking is at least makes sense, okay? Maybe it's not the correct one, but it makes sense. So now we want to understand how well does the mapping agrees on the test genes. So the genes that are not the 1000 that we use for training in this case. So we held out a lot of other genes for testing. And on test genes, you can see that the agreements with the predictions increases with the density of the genes that I have expressed, where if I increase the sparsity of the data, so if here you have genes that are very poorly expressed in the reference dataset, which is Visium, the dataset which is Visium, the agreement will be low, okay? So here I predict a certain spatial distribution, which is very dense here, etc., but in Visium, this data is really sparse, okay? And so the agreement is really poor, okay? So what we did is to plot all the test genes that we had, so which were the majority of it, because we have 30,000 genes and we use just 1,000 for testing, and plot the spatial correlation with respect to the sparsity in the original Visium data. And we divide the space into regions. So we selected kind of arbitrarily this 60% threshold as a good prediction. Okay, so if the spatial overlap between our prediction and Visium is more than 60%, we call it a good prediction. Okay, that's the arbitrary threshold that we selected. So all the predictions in this region here are good prediction in agreement, okay, with Visium. And mind you that these are test genes, so genes that we have not trained on. Okay. In this region here, we have, they have low sparsity and low correlation. These are the genes in which the model fails, okay, entirely. These are not predicted well for whatever reason. In this region here are the very sparse Visium genes, okay, that they have low spatial correlation. So this is kind of the uncertainty region in which the model performs badly, but we don't know if it's because this is a bad prediction or because the gene was poorly measured on Visium, okay? Because as I told you at the very beginning, the special transcriptomic techniques actually suffer from these dropouts genes, which are measured with sparsity and maybe completely skipped sometimes, etc., etc. So what we do is the following. Basically, we validate it for the ones that they do, for the genes that have some overlap with MERFISH, okay? So for example, here you have a gene, this PLX, weird name gene, okay? I'm not gonna even try to pronounce it, that from the mapping on Visium, so the mapping that we predict is that this gene is gonna have a very high density in the middle. This gene is kind of basically completely lacking from Visium. So the data that we have on Visium is very sparse. But then if we look at MERFISH, okay, on which this gene is also present beyond that 254 ones, we see that the expression is concentrated in the middle. So again, this gives us confidence that our prediction is kind of the correct one, and this is an artifact of Visium having this gene completely dropped out from the distribution. Again, you can see it with this other gene, okay, which is predicted to have a high concentration in the top part, which is the same thing that you can measure on MERFISH, but in Visium, this is a really sparse thing. So it's measured in some spots, but in some other is completely dropped out, okay? Another way that you can validate it is now that we have a way to map on two different types of data, so both in situ, so MERFISH and spatial transcriptomics, Visium, you can perform the mapping simultaneously and see if the mappings coming from two different techniques agree or not. So if I map the data using MERFISH as a reference, and if I map the same data using Visium as a reference, if the two mapping are kind of consistent, I have a good chance that the mapping is correct, okay? Makes sense. And you can see that actually in these genes that, again, are very sparse on Visium, okay, so they are in this region of uncertainty in which the model performs poorly, but we do not know if it's because of our model or if it's because of Visium being sparse and like having a lot of dropouts. The mapping computed on MERFISH and on Visium kind of agree. So this predicts a high concentration here, this predicts a high concentration in the middle, this predicts in the top, and et cetera, et cetera. You can see that that kind of consistent. Okay, this convinces you, basically, or at least it was convincing enough for us, to claim that in this uncertainty region, basically what we are doing is we are kind of correcting from dropout genes of Visium, and that's why they are not matching well. Okay, it's because this is the region in which Visium has a lot of dropout genes, so and it doesn't perform well. This region is completely wrong. Okay, still. Okay, so this concludes this part on mapping into space. So if you have questions, then I'm gonna move on. Do I have time? Yeah, I have some time on a slightly different stuff. So basically, we were able to build this nice pipeline that like took these three different type of data, so single cell RNA sequencing, Insitu Techniques, and spatial transcriptomics, which all had their drawback, and we found a way to kind of harmonize them and map them into space in a way that we can have the whole transcriptomics at a single cell level in the common coordinate framework, okay? And that was the missing part that we needed for the mouse brain atlas. Again, we have to translate all these things to human, which is a huge, huge endeavor. So it's not just a technical stuff, but still it's a first step. Okay, yep. Yeah. Not necessarily. No, not necessarily. So yeah, exactly. So again, the thing that we extract this cell density information from is the reference atlas. I'm going back. Blah blah blah. Where do I have it? So is the reference atlas, okay, which is the average mouse brain. Okay, so each mouse will have a slightly different brain, but again, we want to construct like an atlas, which is like a model, okay, of how like your organs will be placed slightly different than mine, okay, inside the body, but roughly like we have the heart more or less in the same position. And if you have to build a human atlas, we have to decide on a place to okay. And even the experimental image that we used are actually taken from different labs in different moments and with different techniques. Like you can see the stain used to take the histology is different, everything is different. So yeah. Okay. So this concludes this first part, which was the work that I introduced at the beginning. Then we had another work which is not really related, so it was a completely different stuff, but I think you can see that kind of the inverse problem of what we just explored. So what we explored is a technique that allows you to take single cell data, okay, so single transcriptomic data and single genes, and map the single genes into space and into the histology. Okay, now what we're trying to do is kind of invert the thing. And from histological image, we are trying to predict single genes. Okay, so I'm going to introduce the biological setting first. This is going to be quick, so don't worry. And then the deep learning stuff that we use. But just to be clear that this is a completely different work. Okay, the two are not related. We do not use the same mapping algorithms. But I think that it's kind of, you can think about it as kind of the inverse problem of what we just did. So let's start with the biology introduction. So I think you have all heard about CRISPR-Cas9, okay, these techniques by which you can modify the DNA at a single like gene resolution, okay? What people are able to do is basically they can start from a blank cell, which they call human wild type, which again is kind of the prototypical human cell. So a healthy cell, average cell, which is real, okay? They have it in vitro, okay? And then start adding mutation one to one. So each one of these letters means one single mutation in the genome, specific one. And this healthy human wild type with this mutation will evolve in different types of melanoma. Okay, so all these CBT3, CBTP, blah, 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 these cells here are all melanoma cancer cells, okay, of different types. So some of them have a darker pigmentation, some of them are more aggressive, some of them are less aggressive, etc., etc. So all the thing. And the cool thing is that, again, if you do this mutation, so you do the single cell RNA sequencing and you map the manifold of the genes into the two-dimensional space, you can actually make out all the single genes mutation that they do from the human wild type. So this is the WT is the wild type. They add the C mutation, then the CB mutation, and when they add the T mutation is the jump from the healthy cell to the cancer cell. Okay, so all of these are healthy cells, more or less, with some mutation. Now you have a cancer, okay? And then you can move in the space of cancer cells, adding more and more mutations. So CBTP, CBTP3, CBTPA, and each one of these letters is an additional mutation each time, okay? So this is what biologists did, okay? And then since biologists are bad persons, they pick these cell types and they put them on immunodeficient mice, okay? And they let them grow the tumor, okay? And so you can see here different tumors have different like physiological features. So you can see that this tumor has a lighter pigmentation. This tumor has a darker pigmentation, as like the first thing that you can see. Maybe these mice died faster than these other ones. I don't know, but they had a list of like different pathological features that they were looking at. But the interesting thing is that then they were able to take a biopsy of these tumors and have histological images. And in the histology, you can still make out some of these physiological differences that we were talking about. So some types of tumors are more dense, cells are a bit more organized or disorganized, stuff like that. So the question that we asked is, since we have a very, very clean data set, because in general, like tumorous cells in wildlife, in humans, have a lot of mutations. Okay, you have the mutation that causes cancer, like the specific one, but you have a plethora of other ones. So you cannot count the single mutation that actually originated that feature. Okay, in this case, since they built this thing from scratch, going from single mutation, we had the chance of having a very clean data set, okay, in which we actually knew for each one of these images the exact sequence of mutation that caused that particular type of tumor, which is nice. And so we asked ourselves the question, can we train a classifier to recognize the genotype of the tumor, so the mutation that happened from looking at the histological image? And if so, is this any useful in predicting like something on real cancer? Okay, which will be the follow-up question. So first problem, real world problem. These histology images are huge, but like huge, okay? I could not load more than one image at a time in the RAM, okay? And I had like, I don't know, some 100 gigabytes of RAM, so they are huge, okay? So training becomes a problem. And what we did is that we took the histology image, okay, and then we subdivided it in 2048 times 2048 tiles, which for the ones of you who work with images, okay, in machine learning, you know that this is still a big image to process, okay? I mean, MNIST is 28 by 28, so it's a piece of cake, okay, with respect to this. And these are RGB also. So we extract, we make a tiling over all the tumor. Of course, we do exclude the section which do not have enough tumor inside to be considered of any interest. And then we subdivide this tile in overlapping patches. So we take these patches, which have some overlap with one another, and we end up with like patches of 512 times 512, and we end up with a number of them, which I do not remember, like n patches of RGB 512 times 512 images, which are kind of a size that we can manage to fit into a GPU memory and train in batches. Basically, that was the requirement that we had. Then we took this encoder, which we took it from the winner of this competition, which was a competition on breast cancer. Okay, so it was a network which was already trained into histology images of breast cancer. So our hope was that it was already somehow able to encode the distinct features of histological images, more or less. Okay. And then, and this was the structure. So it had a bunch of convolutional layers with batch normalization. And then in the end, what we got is it was a number of feature maps of dimension 64 by 64. Okay. So the results, so all these works that I'm going to tell you, this deep learning model is from this work, which is ours. And then we take the feature maps, okay, extracted in such a way. So for each tile, so remember, from the histology image, we take the tile. From the tile, we take the patches. And for each patch, we extract the feature maps, okay? And we take the feature maps, okay, and we feed them on this CNN architecture, which is what we actually train, okay, in this pipeline, which is again a bunch of convolutions, okay, and then in the end we deconvolve the whole thing with a one-by-one convolution, and we apply a softmax, okay, at the end. And what this tells us is just the prediction, a probability distribution on what he thinks the most probable mutation is in that patch, okay, that he's looking at, okay. And then on a single patch, we have the training label, we have the true label because we did the mutations, okay, on these images, so we know what is the correct label of that patch, and we can compute the cross entropy between this propagation, this prediction and the true label, and do the back propagation and train this network, okay? So we did all of this. I do not remember the exact number of stained image that we had. I can find it later if you're interested in, but it was not huge, okay? It was kind of limited. But it was enough to train, at least on this, since we did not have a huge variety of gene mutations, so the number of possibility was pretty limited. And then what do? We train a network, and at the end of the training, we can use the output to predict the patch genotype. From the patch genotype, we can have a majority voting procedure to decide the genotype of the tile. And for each tile, we can predict, like, the genotype of each tile, and from the genotype of each tile, we can do a majority voting or a consensus procedure to decide the genotype of the whole histology image, okay, and generate a prediction, okay. So here you sum, okay, the procedure that we did is to sum the probability of across all the tiles, and that will give us a new probability vector for the whole, like, section, and then we took the max and predict the whole tile. So this is what the output of our model looks like. So each one of these is a tile, and if the confidence was below a certain threshold, which I do not remember, we did not assign the genotype. Okay, so if the output of the softmax was not above a certain threshold, we did not assign, and you can see that in most cases, like edge tiles were not assigned because they didn't have enough information, apparently. And we have a lot of cases. So we have some cases in which the prediction is really, like, the consensus is really high. So in this case, our algorithm is really convinced that this tumor, this mouse tumor, is a CBTPA1, okay, is caused by this mutation. In this case, it cannot really decide anything. It's all unassigned. There is apparently not enough information for it to kind of call a genotype. And in this case, it's very undecided. So for some of them, it calls CBTP. For some of them, it calls CBTP3, and some are just unassigned. But all in all, okay, on the test set, the predictions that we got were pretty good. So you can see the area under curve. So the ROC characteristic curves look pretty good. The worst one are the CBTP and the CBTP3, okay, CBTP3 ones, okay, which are a bit lower than kind of ideal. And you can see on the test set that we are able to call like with high confidence a lot of the types, and CBTP3 and CBTP3 are the ones which that we have a bit more difficulty discriminating between one another, because the most of the time CBTP was confused for a CBTP3 and vice versa. Okay, so for some reasons the pathology features are not really distinct enough from these two species to make out. Yeah.
So when you don't go past the test.
Yeah. Yeah.
There are biological investigations are related.
Yeah.
You see that you make it
okay, so we try. So I don't know, because I know not. So I look at these images and I say, oh, a nice blob of stuff. Okay, I have absolutely no idea of how to make sense of it. Exactly. So we try to discuss a bit with Aaron and Elena, which were the lead biologists that did this work. And they kind of like, for some cases, they were able to kind of say, oh, yes, maybe this region is too dense. So I don't have a way of showing you, but there are some tumors that have very weird. So here you can see all these like white shapes or regions that are very deformed and stuff like that. So in most of cases, they were able to kind of find a justification of like the type, oh yeah, these regions isn't, because then when tumoral growth is not really nice, even growth, etc., it's very chaotic. And sometimes you might have like regions of good tissues inside the tumor. You could have some like, you have all sorts of weird stuff. Yeah. We didn't actually look at that. So if it was below a certain threshold, we kind of discarded it because it was too noisy otherwise. Yeah. And most of the time, the cases in which it did not meet the threshold were either edge cases in which even the biologists were saying, oh, this is an edge, I don't really care about it, or were cases really weird of these like good tissues embedded into tumor tissues, etc., etc. So again, these are the very good results on our test set. You see that since the images were taken in one lab by a couple of persons, okay, we do not have a lot of images to test on. So this below is the number of images that we tested on. So here we got 100% of 5 images, which is good. It's still 100%, but I mean, it's not that much of a test set, but that's what we got. Again, this is the problem when you get to work on real data. You do not have all the data that you might want in the world. You just have the data that the lab could take with their means, and that's what we got. Nevertheless, we had high confidence in saying that for this human melanoma grown on mice, okay, we had a good way of predicting which mutation occurred, okay, with kind of a good confidence. Then we had the follow-up question, which was, does this correlate with real human patients? So these are human melanomas grown on mice, okay? So does this have any correlations with human melanomas grown on real patients? Luckily enough, we have plenty of data in this case, because this is this huge data set, which is called TCGA, okay, which is a data set of human tumors and stuff, and there are a lot of melanomas as well. And for a lot of these melanomas, there is also attached to this database a lot of genetic information that they sampled from the tumor. And so, for example, here the situation is a bit more complicated because now, since, as I told you before, in humans, we do not have a specific mutation, but we have we can see that a certain pathway, they call it, so a genetic program is activated. Okay, so maybe the mutation, like the P mutation, activates a cascades of event, right? It expresses a protein which activates a certain function on the cell, and the cell activates another thing. And so a very complex chain of event, which brings to the actual histology feature in the end. And what we looked at are these programs, these genetic programs, which are correlated with the mutations the specific mutation that we did on mice. And we could see that for some of them, so these mutations are correlated, this OXPHOS pathway is correlated with, if I remember correctly, the CBTP CBTA and CBTP mutations, if I recall correctly. We can see that here we can predict with some confidence, okay, it's not really high, okay, it's not a receiver operating characteristic that goes to 1 as the one that you showed before, but is better than random, okay, it shows some correlation with the human tumor. For other genetic programs which are like activated by the other mutations, so the 3 mutation, the T mutation, etc., we do not observe like significant correlation. So it means that this expression of features in human is very complicated, okay? It's originated a cascade of events and is not sufficient to have one mutation on a mice-grown tumor to infer the precise mutation. Nevertheless, the fact that with this data set, we got some correlation of a genetic pathway is kind of promising in a way that we have the hope of somehow with enough data, with enough like carefully selected and trained data to build a model that looking at an histology can tell you the exact genetic mutation that caused that tumor, which is huge, like from a clinical point of view, okay? So to conclude, using machine learning in biology, we have been able to relate genes to anatomy, so to histological image, extend gene throughput of previous spatial techniques, genomic techniques, and increase the spatial resolution of some others that were able to have a high throughput but low spatial resolution, and also to identify single mutations from the histology image. So that's it. Thank you for bearing with this biology class. Yep. The algorithms are in the paper. Yeah. And also the details of the like more technical, like batch size, etc. So this one, I do not remember if we open sourced it. The one of Tangram and the Siamese neural networks, etc., are all like we have the source code on the broad GitHub. Yeah. This one I don't remember. Because in the end, I was discussing my PhD and I kind of left the project.